I didn't think about that at all, so I will mute.
Testing, testing. Okay, that works just
Okay, I've got the green light here. I think our tech is up and running, so we're going to get going. I would like to introduce Emily Tarsa. I want to tell you just a little bit about her. I know many of you know her quite well, but she graduated from SUNY ESF in 2013, and shortly after landed in Ogden, Utah. She was at the Ogden Nature Center as an intern for a while, working in habitat restoration and environmental education, and then she was their volunteer coordinator. I first heard from Emily in about 2015, and sadly, I didn't have any funding for her at the time, but she was persistent. And by January 2017, we got her in the lab, and the rest is history. So, you know, I like to think some people are worth waiting for. As a faculty member, you take on students, they have great CVs, great reputations, but you never fully know what they're capable of. And it has been just a gift to work with Emily. So, and it's been quite a few years. So we've had a good journey. Sorry. We've been to many wetland meetings. We had an epic journey to South Africa. We took an incredible trip across the deserts of Nevada, collecting seeds, mass gatherings in the pandemic in my backyard. It also seems like life has tried to thwart her efforts along the way. We have a new line in our safety document because of an interaction that Emily had with our lab door and her finger. Her first spring in 2017, we knew that literally every Tuesday and Thursday there would be an epic snowstorm, because that is when Emily would be driving up from Ogden, and she would arrive, she'd get there, but she looked like she'd been through a massive snowstorm. So car accidents, baby, young children, pandemic, COVID multiple times. Nevertheless, she persisted. And I think, you know, Emily is a hugely accomplished scientist. She got two fellowships that I want to recognize. One is the Garden Club of America Fellowship in Ecological Restoration, as well as the really prestigious Ducks Unlimited Canada, very generous fellowship that she got over multiple years from them. She was also awarded our college's PhD Researcher of the Year just last year. And one of our committee members commented that her comprehensive exams were the best they'd ever seen. So she is now a faculty member at Georgia Coastal University and has been teaching classes already for a few weeks. So we're lucky to have her here, and also lucky that she turned her attention during the PhD to addressing a really pressing challenge to wetland restoration in North America, and particularly in Great Salt Lake wetlands. Our wetlands are globally renowned for their avian habitat and face invasive species, droughts, water diversions, and how can we overcome these efforts? How can we restore native plant communities, diverse native plant communities? And the key that Emily has looked at is about the seed and seedling ecology of native species and how that can really help guide wetland restoration. So I will now turn it over to Emily, and I will give her the microphone.
All right, thanks, Karin, for that introduction, and thanks everyone for being here today. I'm excited to finally be able to share the results of what I've been working on for the past 6 years, looking at seed ecology and regeneration processes to inform seed-based wetland restoration. So to just kind of set a picture of where we're going today during this talk, so I want to start by introducing the system that we work in and some of the challenges that are unique to that system, as well as other wetlands across North America. And then move into these 3 data chapters looking at native and invader plant interactions, functional trait responses driving early regeneration, and then modeling demographic probabilities and clonal production. And then finally just wrap up with conclusions and kind of management takeaways and scientific implications. So I'm sure many of us are familiar with Great Salt Lake wetlands. As Karin mentioned, these are globally renowned wetland complexes because they sit right along the Central and Pacific flyways in this really Intermountain West region. And so because of the position in the landscape and the fact that we're in a really arid region, these wetland complexes are significant for millions of birds that move through the system for breeding, resting, foraging, as they move along these migratory pathways. And then even on a local scale, the Great Salt Lake wetlands are really important. We only have very limited wetlands here in Utah, obviously, and 85% of those are located around the Great Salt Lake. So the wetlands around this area provide a lot of recreation opportunities for waterfowl hunting, obviously migratory bird habitat, and a number of other important ecosystem services and functions. Oh, you're good, thank you. So wetlands are highly prone to invasion, particularly invasion of Phragmites australis. And they're prone to invasion because of their position in the landscape, right? So oftentimes they sit along the bottom of watersheds. And so we see a lot of high nutrient input, high propagule input into these systems. And Phragmites is a really common wetland invader across North America. So we can see here, Phragmites in the Great Lakes, in the Chesapeake Bay, in the Adirondacks. So it's a really prolific invader across the United States. And it's no different here in the Great Salt Lake. So Phragmites has relatively recently invaded this system. It was first identified here in the early 90s and has since spread to tens of thousands of acres across Great Salt Lake wetlands. And you can see from this picture, it's incredibly dense. It forms these monotypic stands. It outcompetes a lot of the native vegetation. That's the foundation of a lot of those important functions and services that we really care about. And Phragmites has a number of different modes of production. So it reproduces vegetatively, but then we also have thousands and thousands of seeds that are produced on every seed head that we see here. And so seeds are a really common form of reproduction in canopy gaps and disturbed areas. We see a lot of regeneration by seed from Phragmites. And so because of this kind of really dense, impactful plant, managers and researchers have spent the last decade or so trying to identify effective treatment and control techniques. A lot of this work came from Dr. Christine Rohal, a former PhD student, as well as Chad Craney, a former master's student and current manager on Great Salt Lake. But they identified this 3-year cycle of herbicide spraying, mowing, removing all of that biomass. And when repeated over 3 years, it was really effective at reducing Phragmites cover, reducing the number of seeds in the seed bank, and also starting to kill that rhizome mat. But unfortunately, after Phragmites has been treated in a lot of these areas, we end up with wetlands that look like this. So there's not a lot of passive recolonization in previously invaded Phragmites wetlands. And a lot of this is really context dependent. So there are situations where we do see native plants regenerating. And a lot of that has to do with the length of time that Phragmites has been on the site or the surrounding vegetation. But we do see areas, where Phragmites has been there for a long time, we remove it and the seed banks are so depleted that these native plants aren't returning, or when they do return, it's often these annual native weedy species and not those perennial habitat forming graminoid species that managers really care about. And this happens in Great Salt Lake wetlands, and I posted a bunch of other studies in wetlands across North America where they see the same problem, the seed bank depletion after treating Phragmites. And so this leads us to investigating active revegetation approaches to restore these native plant communities. And my research focuses specifically on revegetation through seed because there are numerous benefits. So it's less expensive and less labor intensive to install, especially when you're working over large areas like that in the Great Salt Lake. And then you also have this added benefit of increasing the adaptive potential of your plant community. So because it's more feasible to collect seeds from multiple different locations, you end up with a seed lot that has different genotypes that say, some of these genotypes can do better in hot and dry conditions. Some of these genotypes might grow taller and be more competitive. So ultimately that leads to building these more resilient and resistant plant communities. But there are a number of management and ecological challenges to seed-based restoration in wetlands. First is that there's limited guidance available for seed-based restoration in wetlands. So the traditional approach has just been to allow these systems to naturally recolonize on their own. As that is no longer successful in a lot of these wetlands. It's now kind of turning to how can we actually apply seed to successfully restore these systems. But to date, there just isn't a whole lot of guidance on how exactly we do that. And then in wetlands, we see a lot of spatial and temporal abiotic variability. And this is true, especially in Great Salt Lake wetlands, where we have these water diversions, water challenges. And so you see a lot of variation. And so there's less information on how these seeds and seedlings are responding to those different conditions. And then another big challenge is we see high mortality in early plant life stages. And this is across the board, you know, seeds, seedlings, that's the most vulnerable point of a plant's life cycle. And so in upland seeding, for example, they see about 90 to 95% mortality of the seeds that are introduced to a site. So really low success rate in a lot of cases. So moving on to kind of the foundation of my work. So I'm really interested in the drivers of recruitment outcomes during wetland community assembly. And when we think about the realized plant community, or the community that we see from assembly processes, we can think about differences in structural composition of the vegetation. So invader cover and abundance, for example, and our native cover and abundance. And then we also can see differences in functional capacity of that realized wetland plant community. So the ability for those native species, for example, to resist future invasion, and the ability of that species to be resilient to changes and disturbances. And this process of building this realized plant community is the result of a lot of other interactions beyond that. So we have biotic influences, such as the native sowing density relative to invasive propagule pressure, the functional traits or characteristics of seeds and seedlings themselves. And then we have the interaction with these abiotic influences. So specifically, we can think about nutrient availability, water availability, and temperature regime. And then finally, with these biotic and abiotic interactions, we also have this temporal component, or this temporal dependency. So we can think about the order of plant arrival, for example. So when some species arrive to a site versus when others do, and that's called priority effects, which we'll talk a little bit about later. And then we can also think about the specific transitions that happen in these early life stages and how that changes over time, and how these biotic and abiotic influences change those different transitions in the early life stages. So moving on to this first chapter, I looked at native and invader plant interactions across sowing density, propagule pressure, and abiotic conditions. So that's specifically looking at these components in our conceptual model. So the main research questions driving this chapter was what native sowing density maximizes native recruitment while minimizing Phragmites emergence from the seed bank? How does the abiotic context, so water and nutrient levels, alter these native to invader dynamics? And then how does the order of arrival or priority effects influence plant community outcomes? And specifically, like if we think about what questions this is addressing from a management perspective, it's more of the how of restoration. What native sowing density, where should we sow, when should we sow? So these are the species that we worked with in this study. We had 5 native species that were pooled together in a mix based on the recommended sowing rate that's currently applied in Great Salt Lake wetlands. And then of course we had our invader Phragmites australis. And so we had a number of different experiments addressing these questions. So 3 total. We set it up in this split plot design. So we set up these large wetland mesocosms from these kiddie pools. They were subdivided into different quarters. So that was kind of our split plot unit. And we manipulated Phragmites propagule pressure, so the number of Phragmites seeds that were there in the seed bank. We also manipulated native sowing density. So we had our recommended rate that's currently applied in Great Salt Lake wetlands, and then 2, 3, and 5 times that rate. And then we manipulated hydrology and nutrient levels. And then we also manipulated sowing timing. So that was looking at those priority effects specifically. And in this treatment, no native priority meant that Phragmites and native seeds were sown at the exact same time. Our 2-week priority meant that native species were sown 2 weeks earlier, and then our 4-week priority is 4 weeks before that. And it seems kind of intuitive that when we sow native species first, we're going to have more native species in the plant community, but there's actually a lot of question about whether or not that's actually borne out. Because in some systems, under some conditions, priority effects might not benefit native plants at all. And because invasive plants are often incredibly competitive, even at these early stages, no matter when you sow, Phragmites or your invader can take hold. So priority effects themselves might not matter. And so that was kind of the big question here. In terms of data collection, so we went through and collected percent cover data every week during the experiment. And then we pulled our above ground end of season biomass by species. And in our models, we looked at the vegetation response variables, which was Phragmites australis or our pooled native. We looked at percent cover and end of season biomass. And we modeled this in a randomized balanced split plot mixed effects ANOVA. So our random effects here was our pool as our whole plot factor. The quarter that was nested in pool was our split plot. And then because of the repeated measurements of native and Phragmites within the same quarter, that was our split plot factor. And then our fixed effects, depending on the experiment, we had Phragmites density, native density, and then our treatment, which was either water times nutrients, or we looked at sowing timing. And then after these models, we ran these 2 key post hoc comparison tests. We look at the contrasts for significant effects of the model to kind of pull out these significant patterns. And so a number of interesting findings that I just want to point out here. So we found in this plot, we're looking at Phragmites seed density on the x-axis. So we have 0, 500 seeds per meter squared, and 5,000 seeds per meter squared, which this 5,000 number is the average density that we see in Great Salt Lake wetlands themselves. And then on the y-axis, we have percent cover, and we're looking specifically at our native percent cover when it's sown at 3 times the recommended rate in blue and 5 times the recommended rate in orange. And so we can see here from this plot that the highest native sowing density, that 5 times the recommended rate, resulted in significantly greater native cover when there was a lot of Phragmites seeds in the seed bank. So there is some evidence to support increasing native sowing densities in order to maximize native cover. But we did find that the biggest driver of plant community outcomes was related to Phragmites propagule pressure, so the number of Phragmites seeds in the seed bank themselves. And so here we're looking at our native response in blue, our Phragmites response in orange, percent cover on the top graph and biomass on our bottom graph across Phragmites seed densities. And so we found this really critical threshold at 500 seeds per meter squared. When there were less seeds in the seed bank, we saw much more native cover and native biomass. But of course, this changed quite a bit across abiotic conditions. Interestingly, we didn't find any support that nutrients changed these dynamics, but low water conditions resulted in greater Phragmites suppression relative to high water conditions. And so here we're looking at Phragmites cover on the top, Phragmites biomass on the bottom. The high water is in blue and the low water is in orange. So you can see, especially as we increase native sowing density, we get much more suppression in the low water treatment. And this was largely driven by one species. This is Distichlis spicata, or saltgrass, which is a drought tolerant native species. And then we did find benefit for sowing native seeds earlier in the season. And it yielded a much more native dominated plant community. So this 4-week native priority is when native species were sown 4 weeks prior to Phragmites versus 2 weeks versus them being sown at the same time. And interestingly, we saw this benefit of Phragmites or priority effects regardless of the sowing density. So it was the same pattern at the lower sowing density versus the higher sowing density, which suggests that in situations where resources are limited, applying this early sowing technique can be a benefit if we can't necessarily increase sowing density. And again, this was driven by one species. So this was saltgrass. And so the big management takeaway from this chapter is that site prioritization is important. So at the site level, we want to identify sites that have low invasive propagule pressure in order to maximize native outcomes. And then from a landscape level perspective, this is a really important finding to push the need for including propagule pressure management into our management and restoration plans. So thinking about ways that we can cut down on sources of those propagules in the landscape to reduce dispersal and reduce overall propagule pressure. We also found that increasing native sowing density does promote higher native cover and biomass, and that where possible, combining these water drawdowns with these drought tolerant natives in the seed mix like saltgrass was effective at suppressing Phragmites. And then incorporating early season species into the seed mix and sowing earlier in the season to kind of induce that priority effect advantage. But of course, this is contingent on species growth requirements aligning with those early season field conditions. So next, we're going to talk about the third chapter, functional trait responses driving early plant regeneration in wetlands. So here we're looking at these functional traits or characteristics of seeds and seedlings as our kind of biotic influence, and then its interaction with water availability and temperature regime. And so here, we previously talked about how we perform seed-based wetland restoration. And the goal of this chapter was to really understand why some seeds and some species perform well in restoration and others don't. In order to get at that question, I took this functional trait approach. And so functional traits are morphological or physiological characteristics that impact plant growth, reproduction, and survival. And so there's a lot of research on plant functional ecology for adult plants, and we can think of some of these functional traits like plant height, which has implications for how plants compete with one another. We can look at root length and structure. We can look at parenchyma development. So that's an important adaptation in wetland species that allows for increased gas exchange when conditions are flooded. And then we can also think about the number of seeds produced, which has implications for reproduction. And the benefit of using functional traits is that it can be this common currency. So it's a way that we can move from species-based recommendations to more of these trait-based predictions, right? So in the last chapter, we talked about how saltgrass, for example, was a really effective competitor, but saltgrass isn't going to be a good competitor everywhere because it doesn't grow everywhere. So the purpose here is trying to find what exactly is it about saltgrass, about the seeds and the seedling themselves, so that we can apply that to other species and other wetland systems. And so in terms of functional traits, plants exhibit strategies and trade-offs in these traits during growth. And so decades of research has identified this really common pattern in plant development such that functional traits are kind of constrained along this resource gradient. And so this is often observed as this continuum between conservative growth traits and acquisitive growth traits. So on the conservative end, you have plants that have really low specific leaf area and specific root length, low growth rates, and really high biomass allocation. So this looks like plants that have really thick leaves, thick roots, they grow really slow, they're really drought tolerant or tolerant to herbivores. And then on this other end, we see this acquisitive plant strategy where we see high specific leaf area, high growth rates, low biomass allocation. So this is that more of that like fast and cheap growth strategy that we see a lot in annual species. And so we can use this information to predict where and how plants grow, and also how plants respond to environmental changes. So as I mentioned, like the conservative end, these are more drought tolerant species versus more drought sensitive species on the acquisitive end. But there's still a lot of unanswered questions in terms of using functional traits during regeneration. So a big question is whether or not seeds and seedlings operate along this similar spectrum and this kind of predictable spectrum in the same way that adult plants do. And there's a lot of mixed findings of this to date. And then we also don't really know if these same regenerative strategies apply to wetland species. So much of the research to date has been focused on upland terrestrial species. And then there's a lot of question about how regenerative traits vary between species and then also within species across these populations. And this has implications for species selection, right? We can identify maybe species that germinate more readily than others. And also for seed sourcing, we might be able to identify, populations that are growing in these hot, dry conditions may do better at a restoration site experiencing hot, dry conditions. And so the research questions for this chapter is looking at how do morphological and physiological seed traits vary across species and populations? Do restoration site level abiotic conditions influence germination and seedling traits? And then how do dormancy and germination traits align with regeneration traits across species and populations? And so thinking about the different kind of phases of regeneration and the data that we collected. So we have the process of germination when the radicil starts to emerge from the seed. The traits that influence that we've measured is time to germination and seed mass. We also have this emergence and establishment and kind of survival, this early seedling stage. And we measured a number of different traits that link to seedling growth. So in orange, we have our growth traits. In blue, we have these tissue construction traits. And then in yellow, we have biomass allocation traits. And then we have from survival, we see the mature plant and seed production. This is where much of the functional trait research currently lives. And then beyond that, we have traits of seeds themselves that influence how these seeds persist and disperse in the landscape. So seed dispersal is one kind of trait that we were interested in, or one function that we were interested in. And we looked at seed buoyancy and then persistence of the seed in the seed bank. So the length of time that it can remain viable in the soil. And we looked at traits related to that, such as seed dimension, seed coat thickness, and depth of dormancy. So for this research, we took many massive seed collection trips across the Intermountain West. So we collected seeds from all the way down in Nevada and then all the way up to Montana. So for our 3 bulrush species, we were able to collect multiple populations. Some of the species we worked with only had a few populations along the Great Salt Lake. But yeah, we did a lot of seed collection from a lot of different regions in the Intermountain West. And then we brought all these seeds back to the lab and we did this big seed trait measurement. So in that, we did a bunch of lab measurements to look at seed mass, seed coat thickness. The analysis for our seed trait data, we looked at just linear regressions, looking at the effect of population on the traits themselves. We also did a principal component analysis to identify how these populations and species were structured in terms of their seed trait values. And then we did these standard major axes regressions, looking at the effect of the maternal environment on the traits. We also did a big experiment looking at depth of seed dormancy for those species that exhibit dormancy. So that's this barrier to germination. And so in this, we had a few different replicates, different cold stratification treatments across 180 days total. And we developed this depth of dormancy index from that. We also did this big growth chamber experiment to look at germination and seedling traits. So here we manipulated 3 different temperature regimes based on current and future projected conditions in Great Salt Lake wetlands. And then we also looked at 2 different moisture levels, which we manipulated with polyethylene glycol. And we set up these little baskets and growing containers where we could seed all of these little seeds and then track each seedling individually every other day. We would mark the date that it germinated. We would pull these seedlings either 7 or 21 days past germination. And then we would collect data with this WinRhizo scanner to measure length and area of the roots and the shoots. And then we did fresh mass and dry mass to calculate those traits that we were interested in. And in terms of the analysis, we looked at a principal component analysis, again, to kind of visualize the variance in these traits across populations. And then we took the PC scores from that principal component analysis and did a 3-way ANOVA looking at population and temp and water potential. And then we did these standard major axes regressions to look at these bivariate relationships between the traits of interest. So we found a few really interesting patterns here. So the first, this is looking at our principal component analysis result for our seed traits specifically. And in a principal component analysis, the goal is to reduce the dimensionality of a data set and capture the variance that we see in this data. So along PC1, we captured 63% of the variance in the data set. PC2, we had 21% of the variance. And this trade-off here along PC1 represented light elongated seeds that were less dormant on one side, and then these heavier, rounder seeds that were more dormant on the other end. So this was a really important trade-off that we found in these species. And then if we look within populations, we did find some effect of the maternal seed environment on depth of dormancy specifically. So seeds that were sourced from lower latitudes with higher temperatures were generally less dormant than those sourced from cooler temperature regions. And we also found that seed traits contributed to like habitat adaptability. So for example, we found that seeds with thick seed coats were more buoyant. And so this is likely related to an adaptation to being dispersed by waterfowl. So as these seeds float in the landscape, they're picked up by waterfowl. They move through the gut passage, which is very acidic. So the seed coat thickness could theoretically help assist and maintain germination ability after it moves through the gut passage. We also found that more buoyant seeds had deeper dormancy, which could be this adaptation to water dispersal in the landscape. So seeds are able to hold off germination to allow for water movement away from the parent plant. And this aligned with what we would expect based on the habitat characteristics of our species. So all of our obligate wetland species that are found only in wetlands in these flooded conditions generally had more buoyant seeds with thicker seed coats and much deeper dormancy relative to our facultative wetland species like Phragmites and saltgrass. We also found seedling traits exhibited some important functional trade-offs. So here we're looking at a PCA plot of all of our species sourced from the Great Salt Lake populations. And if we kind of overlay some really important trade-offs or kind of these bidirectional relationships here, we see that on one end, we have species that have fast growing shoots and roots, so high SER and root ER elongation rates of roots and shoots. On the other end, we had these thin shoots and roots, so high SLA, high specific root length. And then we also had this high resource investment to root, so high root mass ratio. And in general, this also kind of followed this pattern where our obligate wetland species had high SLA values, high root mass ratio relative to our facultative wet species that were much faster growing species. And thinking back to this original diagram here, on the conservative end, we would expect, based on what we know about adult functional traits in upland systems, that the conservative slow growing species would have really low specific leaf area versus the acquisitive end, we have really high specific leaf area and high growth rates. But interestingly, in this system, we found that this was flipped. So on the conservative end, all of our plants had really high specific leaf area versus on this acquisitive fast growing end, we had pretty low specific leaf area. So this was kind of an unexpected finding, and it points to an idea that there might be wetland specific mechanisms that are driving plant strategy specifically. So now if we have this slow growing end of the spectrum where we see all of these obligate wetland species versus our rapid growth end of the spectrum where we have these facultative wetland species, we see interesting traits that are related to flood tolerance on this end. And so one of that was our high specific leaf area, which is really unexpected. And this is likely because we have much higher parenchyma development in these species. So we get high surface leaf areas as a result. And then we had some of the expected relationships. They didn't grow as fast. There was much more biomass allocation in these species, and tying it back to seed traits, they had slower germination rates and much thicker seed coats, which is the bet hedging strategy across these really highly variable environments. And in our rapid growth end, we saw that there was much lower SLA relative to our obligate wetland species. There was a high growth rate, and this is likely to escape high water. So we have this flood tolerant strategy and this flood avoidance strategy playing out in these species. And these species had really rapid germination rates and really thin seed coats. So we did find that these traits varied somewhat across species, but less so across abiotic conditions and populations. So for example, looking at kind of these species-specific relationships, we saw that common spike rush or Eleocharis species had consistently higher below ground biomass investment, regardless of abiotic condition. We also saw that Phragmites and saltgrass generally had high above ground growth rate, and that it accelerated in high temperatures. And then finally, for hardstem bulrush, we found that hot temperatures favored this above ground investment in biomass versus our below ground investment in biomass. But interestingly, and kind of to our surprise, we did not see a huge effect of abiotic conditions on the expression of these traits across populations within a species. So this plot here is looking at hardstem bulrush. So after we ran these principal components analysis, we ran a three-way ANOVA with the PC scores themselves to try and identify the effect of temperature and water on these really important axes of variation in the data set. And there were very few intraspecific patterns of seedling traits across conditions. And so this suggests that because we see potentially really high phenotypic plasticity in these species, there's less of a distinction among populations about how it will perform across these temperature and water levels. So this suggests kind of ultimately, and from a management perspective, that it may be less important to collect seeds from such a wide geographic range because the plasticity allows these species to adapt over very variable conditions on their own. So some management and ecological takeaways here. We did find evidence that seed traits can indicate how seedlings will later perform. So light seeds with thin seed coats and shallow dormancy had faster germination time and higher shoot elongation. And then these heavy seeds with thick seed coats and deep dormancy had much slower germination time and high below ground biomass allocation. We also found that this kind of flood tolerance and flood avoidance strategy is likely driving early wetland plant growth, even at the seedling stage. And then we definitely need to further investigate these kind of well-documented plant patterns to assess application to other systems. So a lot of this was originally built and kind of studied in an upland terrestrial context. Clearly, there's some differences in wetland species, and so that same knowledge about how plants will perform doesn't always apply in wetland communities. And then again, seed sourcing across these wide geographic ranges may be less important for wetland species, but genetic diversity of our native seed mixes is still really critical, right? Especially as we have changing climate conditions, ensuring that we have a lot of genetic variation in the seed mix is important. So moving into the fourth chapter, modeling demographic probabilities and clonal production to facilitate invasion resistance. So this is looking here at kind of this temporal component about how these demographic transitions occur over time in relation to water availability and temperature regime. And so the whole goal with this chapter is trying to understand if we can narrow in on recruitment bottlenecks. So if we can identify when seedling mortality is occurring and how the abiotic conditions might change seedling mortality probabilities. And ultimately, the goal here is to try and overcome these bottlenecks and promote these invasion-resistant communities. And so when we think about early invasion resistance, we can think about it as being the net effects of germination probability, probability of mortality, and then the probability and extent of clonal production. So this is the recruitment pathway that was a guide to building our model in this chapter. So we had sown seeds, and then we modeled the probability of transitioning from sown seed to a germinated seed, the probability of a germinated seed dying, or alternatively, the probability of a germinated seed surviving but not producing any clones. And then on the flip of that, we also modeled clonal production in these species. So the probability that a germinated seed would produce 1 to 2 clones, and then would produce an additional 3 to 4, and then 5+ clones. Research to date in general kind of focuses on this early transitionary period: germination, mortality, survival, and kind of our outcome, cover or abundance. But this whole pathway that includes clones is important in these clonally developing species and is often overlooked in research. And so the research questions driving this chapter: how does the probability of transitioning through germination, survival, and clonal development vary across species identity, source population, and abiotic conditions? And then how do species identity, source population, and abiotic conditions influence end of season native cover? So the experimental design and data collection for this experiment, we set this up across 3 different greenhouses. We looked at 2 temperature regimes. So we had kind of our warm baseline current condition that we often see in Great Salt Lake wetlands. And then we manipulated some of these reservoir systems to have hot conditions. So those are our climate projected conditions. We had 2 different water levels, so either 13 to 15 centimeters of water in a reservoir, which led to soil saturation, versus 2 to 5 centimeters as a drought stress treatment. And then within a greenhouse and within a reservoir, we had these containers that were split between life stage side and percent cover side. So on the life stage side, we sowed a known number of seeds, and we tracked germination and mortality and clonal production as a categorical response on that side. And then on the other end, we looked at percent cover. And then we did this across populations. So you can see up in this map here, we had a population from our northern region, a population from our southern region, and then a Great Salt Lake population for our 3 bulrush species. So in terms of the analysis for end of season cover, we ran a mixed effects ANOVA model with a beta distribution, with greenhouse as our whole plot factor, a reservoir nested within greenhouse as our split plot, and our random effect factors. And then as our fixed factors, we looked at temperature and water and species or population, and then ran 2 key post hoc tests and our contrasts. And then for the life stage transition models, we use logistic regressions with a binomial distribution to model each of those individual transitions for every species and population. And so our response variable was the probability of transition. We had our random effects, which were the same as above, and same with our fixed effects. So we found that pre-germination was the most significant recruitment bottleneck generally for all of these species and populations. So despite breaking dormancy in these species, we see quite a bottleneck in terms of the number of seeds that actually germinate in each of these conditions. But of course, this was variable across species and even across populations to some extent. So Phragmites, for example, we saw really high germination and survival probabilities regardless of abiotic treatment. Same with saltgrass, there wasn't a huge effect of abiotic treatment on survival or germination. And then in some of these distinctions, we can see like the one population of SCAC, for example, performed quite a bit better than some of our other populations. And interestingly, so germination was definitely the big bottleneck, but once a seed germinated, survival probabilities were generally pretty high, like there wasn't a lot of mortality. We found that clonality significantly contributed to outcomes for some species. So this is looking at Eleocharis, palustris. And in each of these, so we have date on the x-axis, we have our proportion of seedling clonal production on our y-axis. And then these are our clonal categories. So the probability of producing no clones, 1 to 2 clones, 3 to 4 clones, or 5 or more clones. And you can see for Eleocharis, especially in this high water condition, over time we have no clones produced, but as we start producing clones, that number goes down. So this is a transition graph. But we can see in those high conditions, especially about 75% of Eleocharis seedlings produced more than 5 clones per seedling, which is pretty extensive. And if we compare that to like saltgrass, for example, and Phragmites, each of those produced around 30 to 40%, or 30 to 40% of those seedlings produce less than 3 clones. So there was quite a big difference in clonal production. And all of our other species produce less than 10%, or less than 10% of those seedlings produce any clones at all. And then these species had equivalent end of season cover. So there was a big contribution for Eleocharis in terms of final cover from this clonal production. And then we saw differences in germination probabilities across species and abiotic conditions. So for our 3 bulrush species, we found that high water and hot temperatures increased germination probability. And then for Eleocharis, what was interesting is that high germination occurred at high water, but it occurred regardless of temperature. So temperature didn't really have a huge effect on how it germinated, and it could germinate equally well in high water conditions at cool temperatures versus that hot temperatures. And thinking back to that idea of priority effects, this could potentially be one of those species that can handle those early season growing conditions and can be really effective at suppressing subsequent Phragmites growth. And then we found for Phragmites and saltgrass again that germination was not sensitive to abiotic conditions. We also found that generally high temperatures accelerated clonal development. So for hardstem bulrush, for example, here we're looking on the y-axis is the increased probability that a seedling produces clones. We have our temperature on the x-axis, and we have these different populations at high water levels in blue and low water levels in red. And so we can see that in general at high water levels, as we increase temperature, we get an increased probability of clonal development. And the same was true for Phragmites australis. As we increased temperature in these high water conditions, we had a higher probability that clones were produced. And then we also saw some evidence of population level differentiation in cover and life stage transition probabilities. So going back to this graph with hardstem bulrush and the probability of developing clones, we saw that the Nature Conservancy, so this is a Great Salt Lake population, for example, was able to produce clones at pretty high rates, regardless of water level when conditions were hot. So even at this low water condition in red, as we increase temperature, we still see that increase in clonal production relative to our other populations where there was either a decrease in clonal production or no change in clonal production in low water levels. And then we also saw differences in the probability of germination for 3-square bulrush. So specifically our Great Salt Lake population, Harold crane, had much higher germination probabilities relative to our other populations, regardless of water level. And so some management and ecological takeaways here. There should be a renewed focus on ameliorating barriers to germination, because we found that despite breaking dormancy, that was the biggest barrier to recruitment outcomes. And so part of that looks like finding ways to break dormancy more extensively and in a way that's more logistically feasible for managers, which could also point to potential utility for seed enhancement technologies or alternative ways to break dormancy in these species. And then a renewed focus to manage for wetland conditions that facilitate the highest probability of germination. We found that Eleocharis is a really promising species to facilitate invasion resistance because of its really extensive clonal production, as well as its ability to produce lots of germinating species across temperatures. And then we found that increasing temperatures yields higher clonal production, which certainly has implications for assembling plant communities and climate change, where we might see this species recruitment favored in terms of clonal development when temperatures are warmer. And then also evidence that Great Salt Lake seed collections may provide really important genetic diversity. So moving into conclusions, I just want to kind of wrap up with a few important points. So ultimately, the goal of this research was to move towards this precision restoration framework, where we are maximizing recruitment outcomes in the face of these resources and logistical limitations. So trying to identify very specific mechanisms or points along recruitment pathways in which we can specifically manage to maximize outcomes. So we did find some empirical support for the need to increase native sowing density, the need to reduce Phragmites propagule pressure, and then targeting specific conditions that favor native species. We also identified these really key relationships among regeneration traits that serve as these building blocks towards building more predictive models of seedling outcomes in wetlands. And then finally, we kind of identified the significance of clonality and transition modeling in identifying bottlenecks. So thinking about scientific contributions, we found that functional traits of wetland plants operate in a unique trait space. So this is likely the result of really harsh and highly variable habitats that select for a unique combination of traits at both the seed and seedling level in wetlands. And so we still saw this tolerance versus avoidance syndrome that we would expect from other systems. But this was driven by some unexpected functional mechanisms. We also saw that priority effects, which is a relatively new kind of research frontier in wetlands, is a really promising area for future research. And then continued research on regeneration dynamics is definitely needed, in addition to other tools such as seed enhancements or more extensive dormancy break, and also kind of expanding these studies to increase the phylogenetic diversity of wetland study species. So we had a relatively narrow range of species in this study. So it would be interesting to kind of continue that research and include more species that are, annuals or early growers to kind of see how these relationships change. And then further assessing clonality is incredibly important because it has such an important contribution to seedling growth in wetland plants. And then in terms of management perspectives, we found that the standard application of sowing density is likely not sufficient. So the action there is increasing native sowing densities or sowing earlier in the season. And then species selection itself is really critical. So choosing species that germinate readily in early season conditions, and then prioritizing species with functional seed traits conducive to rapid establishment. So thin seed coats, shallow dormancy requirements, and low time to 50% germination generally led to high growth rates. And then it's important to incorporate a variety of species and sources into potential restoration mixes. And that includes variation in life form and growth history, such as Eleocharis, which produces a lot of clones very rapidly and can provide like another habitat or occupy another niche in the assembling community. And then, of course, incorporating Phragmites australis propagule management into our restoration and land management plans is kind of an important takeaway from this research as well. Before I wrap up, I do want to just take a minute to thank all of the people who helped me get to this point. So first and foremost, I'd like to thank my advisor, Dr. Karen Kettingring, and my committee members, Tricia Atwood, Sarah Knoll, Mark Brenson, and Matt Madsen. And I would like to thank all of the Great Salt Lake wetland managers and all of the other Intermountain West wetland managers that allowed us to come collect seed on their property. Specifically, I'd like to thank Keith, Jason, David, Chad, and Rich. And then I'd like to acknowledge and thank all of the many funders who made this research possible. And research itself can't be conducted alone. And I have had the pleasure of working with some incredible graduate students and lab technicians. I don't know how it's possible that the Wetland Lab gets the most amazing techs, but it happens every time. And this research especially involved thousands of hours of data collection. And I was always so impressed with our lab techs who came in every day, amazingly continued to be there and were so curious and engaged. And then all of our graduate students who helped throughout this process. Finally, I'd like to thank my people, my family, my good friends, my Ogden crew, and then of course, my partner, Greg, who has been incredibly supportive throughout this process. and my two awesome kids who definitely helped me maintain perspective. Yeah, even on the hardest days, they were always there to kind of make it better, and they assisted in the field, and budding wetland scientists here. That's my goal. Yeah, so with that, I am happy to take any questions.
Questions from the audience? And if any of the folks on Zoom have questions, they're welcome to unmute and or post in the chat box.
So early seeding gives saltgrass an advantage. You just left them there, not in a kiddie pool, but out in nature. What would be the next?
So that's a really good question, and definitely an opportunity for further research in this study and in a lot of the research and priority effects. We see this benefit on a short-term scale, but there's a lot of question about whether or not that holds true in 5 or 10 years. So in terms of like the first year establishment, it can be pretty successful, but definitely more research is needed to see if that continues to be the pattern in future years. Yeah.
Great job.
Thanks.
So you did a lot of controlled experiments,
Mhm.
and I wonder what kind of processes out in nature might influence the results from your controlled experiments? Like,
Yeah.
do you expect that those will hold when you actually go out and implement it, or there's some wild cards like, oh, I know this is a deal, but
Yeah.
we couldn't do it.
Yeah.
That always happens.
Right. And that's a really good point. So a lot of the conditions that we manipulated in this experiment were based on conditions that we see in Great Salt Lake wetlands. And the goal of this research was to understand more of those mechanisms. So bringing it into this controlled context helped to kind of tease apart how these seeds and seedlings are responding. But of course, if we applied these in the field, I would not expect the patterns to necessarily hold true. But I think there is a lot of importance in kind of starting from this side and understanding, okay, here's our baseline conditions, or here's this plant growing in these conditions with and without competition. Here is what we would expect. And then we can kind of build up and bring in some of those other conditions that we might see in the field to see how those relationships change from baseline, and then ultimately move towards this more field-based approach where you can kind of, hopefully at that point, you have some estimate of how these seeds and species will respond, and then you can kind of compare the changes that you see in the field to identify other sources of variation or other sources that are driving plant community outcomes in the fields. Yeah.
Okay. Oh, one more. Good job.
Just kind of tagging on with what Peter said, I was wondering if you know anything about kind of either trade-off or synergies between early germination and percent cover and seed production.
Yeah. So in terms of early seed germination, a lot of this was built on some of Ray's work as well. With those annual species that germinate really readily earlier in the season, kind of the trade-off there is they die pretty early in the season. So you see this early senescence. And so for those later germinating species, if that's an invader, then they have this opportunity to take hold. So I think in terms of how to account for that in management and restoration, it's a mix, right? Including some of those early germinating species, as well as some of those perennial, maybe later germinating species to kind of fill in the gaps as those other plants senesce. And then in terms of later seed production, making sure that those perennial species are kind of there at the end of the season to produce seed in that site. Yeah.
More from Peter.
I'm just, this is out of curiosity, are there ways to manipulate the seeds? A nice marinade or something?
Yeah, yeah.
More successful.
Yeah, so in this research, we did break dormancy. And so that's putting all of these seeds in cold stratification, sand and water for 30 plus days to break dormancy itself. This seed enhancement technology idea has been done a lot in upland species, and that is kind of like that, like manipulating the seed coat, including, chemicals that can retain moisture if moisture availability is an issue. There are potentially seed enhancements that can break dormancy, although we haven't tested that in our species. So there are options and opportunities out there, and there's a lot of work being done on that in upland systems, but less so in wetlands. Mhm. Yeah.
Okay, let's thank Emily for an excellent seminar. You see, they're eager to grab coffee and snacks, so if you want a cup of coffee, run and grab, get them now before they come in with their cart and snack it away.